Welcome technicians. Today we look into how to give sample to the our main C311 instrument. Okay, so this is the instrument as it looks. So the top cover I have opened it so that it will be easy for you to see. Okay, so these are all the components. So we have the sample disk, the reaction disk or the blank cells and the reagent disk. Then we have the ISC reagents, cleaning solutions, ISC panel, right? Now, few important things as I told you last time. This is the sample probe. So this is where the water comes for cleaning of the sample probe. This is its home position, and this is where the drying happens. This is the first sample picking position. This is the second sample picking position. That is the inner disk, the inner circle of the sample disk, outer disk of the sample disk, and then it has got a cleaning position at this part, which in the last video you might have seen. Okay, right? So this is the indicator light. So it will tell whether if this light is on, then only we should able to touch the sample disk and all. If it is off, we should never touch or try to manipulate the sample disk. Then we have to look into the monitor. Here it should be either standby or sampling stop. So if these three positions are proper, then only we should turn the sample disk. Otherwise, we should never do that. The reason being, many of the times this probe will be stuck at the sampling positions, but still this access sample disk will be on. But whereas when you look at the monitor, you can see that it will be either in the stop or it will be in a position where red alarm has come in this AU position and all. So because of that reason, the new technician should always learn. So sample probe in the home position, the sampling disk light is on, and in the monitor either standby or sampling stop. These three positions should be fulfilled, then only you should turn the sampling disk. Okay, so this is the sampling disk. So it has got 110 positions, which I told you last time. So it starts with position number 1 here. So this is the outer disk. Complete part of the outer disk is available for sample patient sample, that is up to 55 positions. So 1 to 55 position is totally dedicated to the patient samples. After that, 56 position which goes to the inner. So it goes all the way up to 70. Actually it goes up to 80, but only up to 70 only we give samples. After that we don't give samples. So if you look at this disk, you can make out that after the 80th position, so we have black coloring of this disk, and then we have white covering, and then we have red covering, and then there is a green. Okay, so all these has got a meaning which is very important to us. Okay, so this black color means that these are all the position which is reserved for calibration, means only calibration reagents are put in this position. Patient samples and all should never be put in these positions. Okay, this white background indicates that these are all the positions where quality control samples are kept. Now here only 92 it is showing, but many of the times urine and other things and all, so we reserve the whole 91 to 107 positions as the quality control position. So you should never give patient samples in these positions also. Okay, then you can see that from 99 to 108 it is red colored. Red color means these are all the positions where emergency samples are put. Emergency sample means whatever may be the order in which you might have given the sample to the instrument, but it will always go to the emergency position first and then it will go to other positions. So the 99, so you cannot give one position, two positions as emergency. Emergency positions are only 99 to 108 only. So that is the important thing you need to remember. So from position 1 to 70 position, 1 to 70 position is dedicated for patient sample only. Then 80 to 90 positions are reserved for calibrations only. Then 91 to 107 is reserved for quality controls, and 99 to 108 is reserved for emergency positions. And then this is W1, W2 position, which is for ISC cleaning and all. So that we discuss as we go along. Okay, so now as I told you, what is the importance? Okay, so when you look at this, you might have seen that there are different colors that is present for the positions, right? See, what it means is, see, now in this position you can see that they are all slightly blue colored, right? So what it means is when the sample disk is scanning the instrument, so these are all the positions where it can see that the vacuum containers have been kept. or test tubes or whatever is the sample to be put. So those things are present, like a test tube or a vacuum or a container, or a Hitachi cups. So that is present in these positions, but we have not assigned any test to them. So that is what these blue color tips will indicate. Now white color means here there was a vacuum for which test has been assigned and the test has been completed. After the test completion, we have removed this vacuum. Okay, so that is the reason why it is in this color. Okay, green color means there is a vacuum, the test has been done, test has been completed, all the tests are done, so it is in green color. Okay, now this is red color. S indicates that there is an error that has happened in this particular position. Like if you are given LFT, all the tests might have been done, but one or two tests might have been cancelled, or one or two positions sample clot might have come, or whatever may be the reason. For that reason, this is not completed, so it has come as red color with a indication that the sample, the reagent, sorry, the test has not been completed. Okay, so now we come to the main. Okay, so we learned about the sample disk, the positions, what is those indicators and all. That much you need to know exactly. Now we have come into the main session. So how to give the sample. So please do remember, the Microsoft Word has got a very specific way of indicating the windows. Okay, now as you can see, there are workplace window, reagent window, calibration window, quality control window, utility window, like that. Now which folder is open, you can make out when these margins are clear. Like for example, see calibration, QC and all, it is in ash color. But down when you look at it, this is open only for data review. Now the reason why I am mentioning this is, many of my technicians don't know. They just click test selection, then data review, test selection, data revision, like that they keep on clicking. They don't understand which particular window is actually open. So in this case, data review is open, so there is no this, it is not in ash color because of that reason, and this whole thing is totally open. This whole folder is totally open. So if you select the test selection, see now this data review has come into ash color and it has got a margin here, like how reagent calibration has got a margin, but this test selection, the total color has become clear. So this is how you identify that which particular window is open in the instrument. Okay, this is very important. Okay, don't forget that part. So you should know which window of the is actually working. So that is the thing to know. So don't just keep on clicking, that's it, plus or minus, minus or plus, like that. Don't keep on clicking. Know exactly which folder is in the windows is open. Okay, so now to give the sample to the instrument, so what we do, we go to the test selection. Then we have two options. Either we can give it as a routine sample, or we can give it as a stat sample. If we give it as a stat sample, we can give only disk positions as 99 or 108. Okay, today we will learn about how to give the sample in a routine mode. Okay, only that much we will look into. So you click on the routine mode. The moment you click on the routine mode, immediately, so all these things have become empty, disk position, sample ID, everything. So this is how the new thing looks. So you click on routine, then you select what type of sample you are giving, whether you are giving serum or a plasma, or you are giving urine sample, or you are giving CSF, super, whatever sample you are giving, that particular thing you select. Okay, subsequently this sequence number, please do remember, this sequence number is assigned by the instrument. You should not touch it or change it or do anything about it. Okay, please don't touch the sequence number at all. Okay, now you enter the disposition. So whatever may be the disposition, so you enter the disposition. But important thing is, after the disposition you have to press the enter button. If you don't press the enter button, it will become empty again. Okay, so that means you are telling that at this position, this is the position and this is the position I am entering, like that it will tell. So once you enter the disk position, it will take the position, and then you enter the sample ID. So I have entered the sample ID, and now you select whatever is the test that is required. Sometimes you have to change the sheet because all the tests may not be present in this one window. So you can select this sheet 2. In the sheet 2 also there are many tests are there. So from that also you can pick up. So like that there are totally 5 such windows are there. So based on that you have to take a call. Usually serum only has got sheet 1 and sheet 2. So there is no sheet 3, 4, 5 in our system. But you can in other labs they may be using it. Okay, but in urine and supernatant and all, only sheet 1 is present in our lab. Okay, so now I have taken glucose. Now this has become yellow. So immediately I will put it as a save. Okay, so now I go to the next position. See, I have clicked on the routine. Now this sequence number has changed, so don't touch it. So now again I put the disposition as 37, enter, put the sample ID, and I have selected creatinine and I have saved the sample. Okay, now again I have come to routine. Now in this case I will select urine sample. So the sequence number is 38. I have to put the disposition. So I have put the disposition and then enter the sample ID. Now whenever you are using a particular test, see, many of the times the test value may come very high. So during that time we can give in a dilution mode. See, dilution mode doesn't work for HbA1c and electrolytes. For all other samples you can inside the instrument only you can dilute, like either see you can put it as a decrease means 1:2. Okay, then 1:3, 1:5, 1:10, 1:2, whatever is required. So usually over a period of time by experience you will learn. Many of the technicians will ask me how much to give. So usually I decide many of the times, but remember that decrease is 1:2, then 1:3, 1:5, 1:10, like that. Increasing mode we normally don't give, so you don't have to worry about the increase mode here. So what you do, routine, use whatever is the test, and then whatever is dilution is required, you select that. See, immediately it will give it as, see, it is in decreasing mode, 10 dilutions it is present. Now you make it normal again and then you save it, so that subsequently nobody will have, if you keep it 1:10 and then save, subsequent windows also comes as 1:10 and every subsequent test also becomes diluted samples. Okay, so don't do that. So what you do, routine urine you have selected, then you have put it into 1:10, you have selected the test and then make it normal and then save. Okay, so as you can see, now we have 36 positions, 37 position, 38 position, like these 3 positions we have there. So if you select this particular number, it will become blue and it will tell you exactly what is the test you have selected. Now few things I want to tell you now itself. Please do remember that at 36 positions we have given serum. Okay, now by mistakenly for a sequence number of 37, but I have given 36 position, but I have given urine or supernatant or whatever it is. Okay, some different I have given. So what happens when the instrument is scanning, it will scan that at 36 position I have given serum, but again at a 36 position I have given urine or supernatant, some different one I have given. So when this is a conflict. Okay, now whenever that happens, instrument will give a red alarm and it will give a sample disk error alarm, red, and instrument will totally stop. Whatever test it is doing, that also it will stop and it will totally come to stop. Okay, so please do remember. And at the same time, let us say that at 36 position I have given this and I have given glucose test. Now again by mistakenly I gave 36 position some creatinine or total protein something, and again I gave 36 position only somebody else sample I thought, and I gave it. So what is going to happen is, now since both are serum only, the instrument will do the test, but at 36 position it has done glucose test, but again at the same patient's creatinine also it has done, but the value of that particular is mapped to somebody else because you have given a different number here. So please do remember that mistake. Means at 36 position serum sample of glucose also you have ordered, but instead of ordering it 37 you have ordered 36 position only creatinine, but since both are serum sample only, the instrument will do the test without knowing. But whereas if by chance if you make 36 position one serum and 36 position another urine, supernatant or others, the instrument will identify it as wrong and it will give a red alarm and it will stop. So this is a basic thing which you need to remember. Now after that what I will do, I will give start here. So once a moment you put the start, so this is how it looks like. So you click on the start button. Now whenever you click on the start button, there are two things you need to remember. Since in this case we started from standby, so and we are giving, instead of one number we are giving 35 positions as this thing, so it will ask whether the sequence number is correct or not. Please confirm the sequence number, like that it will give. So since you can give the samples at any positions, there is no need that it has to be in an ascending order, descending order, anything of that sort, but conflict should not happen, that we should know. Okay, so we give OK. Now by chance if the instrument was in sampling stop and I have given, so it would have asked me whether this is an exchange sample or not. So please do remember, always click on not exchanged. Okay, always click on not exchange. If you click on exchange, the conflict happens and the instrument will again come to red alarm and it will stop. Okay, so Once that is done, what the instrument does, that also we should know. So first and the foremost thing, immediately nothing will happen. Okay, it is not like mind ray instrument. So this one will first blink 2-3 times to indicate that you please, it is going to start. Now immediately this sampling disk is going to get locked. Now even after locking, 2-3 times it will move clockwise, anti-clockwise, just to indicate that it is going to move. Okay, so once that is done, first it will slowly move, so that it identifies each of these vacuum positions and identifies where the cup is present, where the cup is not present. So like that it will do. First time it will move slowly, second time it will move fast. Now please do remember, when the first time scanning is done, all the tubings, everything, whatever you have kept, they are all in position. So now when the second time scanning is happening, please don't add any more tubes or remove any tubes. If you do that, there will be an error from first to second matching, and because of that reason, instrument will again give a red alarm and it is going to stop. So when the first scanning happens, so the second tapping, please don't change anything. Means don't change the vacuum. Either you add the vacuum or remove the vacuum. Please don't do it. Now once that is done, from it will show that from standby it has gone to preparation, and then it would have done the sample scan. After the sample scan, it will go for operation, and once that is done, immediately it will come to sampling stop. Now in sampling stop, it will take little time. So normally what happens, so from standby to pick up the instrument, so the sequence are something like that. From the standby, it will go to preparation, and from preparation it will go to operation. It will pick up all the samples, and then it will come to sampling stop. If it is already in the sampling stop, it will give sample scan. So it will scan and then come to operation, then pick up the samples and then come to sampling stop. So this is a sequence, and this is the timing that is required for sequence. And once that is done, these are all the timings it is going to take. For electrolytes, after picking up the sample, it will take 4 minutes. For glucose, it will take only 7 minutes. For all other biochemistry tests, it will take 10 minutes. Whereas for HbA1c, it takes about 14 minutes. Presently, we are not running HbA1c in this instrument. We have a separate high performance liquid chromatography instrument is there, so we are running in that. But maybe in the night time and all, we may have a plan to run HbA1c in this instrument also. So that will take 14 minutes. Okay. So I hope now you might have understood how to give the sample and how to read that values in that instrument. So thank you.